Welcome to the fascinating world of psychiatric genetics, where scientists are unraveling the complex puzzle of how our genes influence mental health. It's a large-scale, multidimensional field that leverages vast genomic datasets to decode the intricate dance between our DNA and behavior. Sometimes amid all this data, our minds can play tricks, seeing patterns that aren't really there, what scientists call apophenia. At the heart of this research are the transcriptome, the full set of RNA transcripts and the tiny single nucleotide polymorphisms, or SNPs, that pepper our genome. These minuscule variations, along with variable number tandem repeats, can influence everything from neurochemistry to brain structure. And don't forget about the non-long terminal repeat retrotransposons, those jumping genes that have been around since our evolutionary ancestors, conserved in regions critical for brain development. Epigenetic changes like chemical tags on DNA also modulate gene expression without altering the underlying sequence, impacting neurodevelopment and CNS function. Think of them as dimmer switches controlling how genes turn on or off, affecting neurochemistry and ultimately behavior. Much of this work involves studying orthologous genes, values conserved across species, to understand human brain function better. Researchers often use transgenic models like mice with modified genes to explore craniofacial morphology and neurodevelopment processes linked to disorders like autism spectrum disorder. When scientists examine gene expression patterns, they look at how transcriptome modulation influences cognitive functions. For example, variations in the serotonin transport gene can affect mood and anxiety. Transcription factors, proteins that regulate gene activity, also play vital roles in shaping brain development. Treatment approaches are evolving too. Cognitive behavioral therapy remains a cornerstone, but understanding genetic variation results helps tailor personalized interventions. For instance, some individuals have heterozygous mutations, meaning they can carry one normal and one altered gene that can impact their risk for psychiatric conditions. Loss of function mutations or null alleles can disrupt normal neural processes, contributing to disorders like bipolar disorder. Many psychiatric conditions are polygenic, involving multiple genes across the genome. Genome-wide linkage analysis helps identify locus-specific chromosome regions that are associated with disease risk, often reaching genome-wide significance. So whether it's decoding the influence of epigenetics, exploring structural variants, or understanding how gene expression patterns shape cognition, psychiatric genetics is an exciting frontier. It's a vibrant mosaic of molecular, cellular, and behavioral insights, reminding us that sometimes the smallest genetic change can have the biggest impact on the mind.